This video is about the Spectronic 20 spectrophotometer. We're going to talk about the principles behind a spectrophotometer. We're going to talk about how a spectrophotometer works, and then we're going to talk specifically about how to work Spectronic 20 spectrophotometer. So first, a very important thing that chemists and other people like to do is measure the concentration of something in a sample. So you might be interested, for example, in the concentration of glucose or cholesterol in your blood serum. You might be interested in the amount of lead in your drinking water, or the amount of mercury in some fish you caught out of some river. Well, one technique that chemists like to use to do that is something called absorption spectroscopy. So spectroscopy just means that we're using light to probe matter, and in this case we're looking at how a sample absorbs light. So in my little drawing here I've indicated that we have a little test tube with some little pink molecules that might absorb light of a particular wavelength. So over here I've drawn this Greek letter lambda right there. That is our symbol for wavelength. And so we know that different wavelengths of light correspond to different colors of light. So the idea of this experiment is we're going to select a particular wavelength where our molecule absorbs that wavelength strongly. And hopefully other molecules in our sample don't absorb that wavelength very well. And so we're going to shine that light at our sample. So that light's going to have a certain incoming intensity or incoming power. So I've labeled that P sub n for our incoming power there. So you'll remember that one way of describing light is it's composed of photons. So high power light or high intensity light will have lots of photons coming in. So if we have a solution that's at relatively high concentrations, we've got a lot of pink dots drawn in here, then those pink dots are going to absorb a lot of that light. And the light that gets through that solution at that wavelength is going to be pretty small. So that's my output power or output intensity. So a lot of those photons get absorbed by the molecules in my sample. That's why this is called absorption spectroscopy. And so I think you can see now why this might be a good technique that relates to measuring concentration. So let's contrast this to a low concentration sample. So in this picture, we see that we have a sample with much lower concentration, so there are fewer dots per unit volume here. We've got the same incoming power at the same wavelength, so we're using the same color light. And what you see is that more of that light gets through the sample. It gets transmitted through the sample. So our output power, our output intensity, is higher. So it must be that something to do with this ratio of output power to input power is related to the sample concentration. And so we will typically define something called the percent transmittance. So we call that percent capital T, percent transmittance. And that's just the percentage of light that gets transmitted through our sample. So we could actually go in with a device that measures light intensity. We could measure the output light coming out. We could measure the input light going in, and a ratio of part to whole. We multiply by 100 to convert that into a percentage, and that's how we would calculate percent T. So that's the percentage of light that gets through our sample. It's the output power divided by the input power, and so we just need a device that measures the output and input light intensity, and that device is called a spectrophotometer, particularly when you're using it in the visible region of the light spectrum, so you're using visible colors of light. It turns out that there's another measure that's kind of useful to think about, and that's the absorbance. And so rather than looking at the light that gets through, we're going to look at the light that gets absorbed. So you might think it's just the difference, percent T, so whatever 100% minus percent T, that would be the percentage of light that gets absorbed, and that's correct. But chemists define a more useful measure called absorbance. Chemists like to use absorbance because it turns out that it is linearly related to concentration. So absorbance is defined this way, capital A, and it is a unitless quantity, although sometimes people will list it as absorbance units, but it doesn't have any units. And the way we calculate it from percent T is it's 2 minus the log, the base 10 logarithm, of the percent transmittance, the percent T. So for example, if you measure your sample and discover that 50% of the light gets transmitted, so only 50% gets through, then we would calculate the absorbance like this. A is equal to 2 minus the log of, since 50% gets transmitted, we would say 50 here. And then when you do this calculation on your calculator, you'll get an absorbance of 0.3. So 0.3 is your absorbance for that particular sample. So that's how absorption spectroscopy basically works. So you might be curious about, how do we know what wavelength to use? Well, normally we want to pick a wavelength that is absorbed by our molecules, but not by other species in our solution. Well, how do you know that? Well, we have to do a different experiment. We have to measure something called an absorbance spectrum. So let me draw that for you. Okay, so what we're going to do is use an instrument to measure the absorbance spectrum. And so what we want to do is we want to measure absorbance, and that's plotted here on the y-axis. So that's just the quantity that we were talking about. Up here, it's 2 minus the log of the percent transmittance, and it's a measure of the light that gets absorbed by your sample. And so what we're going to do is measure the absorbance at a whole bunch of different wavelengths. In fact, we're going to scan through all the different wavelengths. And so when absorbance is high, like it is right here, that means a lot of light gets absorbed at that wavelength. Down here, there's not much of that light of those wavelengths that are getting absorbed, and so this is the light that gets transmitted. So this is the transmitted light, and so whatever colors correspond to those wavelengths, those are the ones that are going to get through the sample and that we're going to see with our eye. So if we see a solution that is blue, it is blue because the blue light is being transmitted. So what color gets absorbed? Well, if you remember art class, it tends to be primarily the complementary color that gets absorbed. So we know, for example, that red and green are complementary colors. So if you observe something as being green, it is because the red light primarily is being absorbed. So in this example, I am using short wavelength light down here, and that's primarily what's getting absorbed. So this wavelength right here is special. We're going to call it the wavelength of maximum absorbance, so lambda sub max right there. And probably that's the wavelength that you're going to want to pick when you're doing a spectroscopy experiment. So you want to see how your sample absorbs at the wavelength of maximum absorbance. That means you're getting the most bang for your buck. So that particular wavelength is absorbed really well. Now there may be other experimental reasons why you might want to select a different wavelength, but typically we will choose the maximum wavelength. And very often when you do an experiment, you will have some literature values that will tell you for your particular substance what is the wavelength of maximum absorbance. But you could also do this experiment using the right kind of instrument. So let's go now and look at how a spectrophotometer is made. That's the instrument that does this kind of experiment, that measures the amount of light that gets transmitted. So here's a little cartoon of the principal pieces that make up a spectrophotometer. So a spectrophotometer is just the instrument that we use to do this experiment that I was just talking about. So first you have to have a source, and I've drawn it here as a little tungsten filament light bulb. So you heat up this light bulb filament and it glows and it gives off all kinds of colors of light, what we call white light. And so we pass that white light from the source through a little slit right here. So we select a little narrow band of that light, and then we're going to pass it through a device that will separate all of the colors of light out. So a prism might do that, but there are other devices that would do that too. I've drawn a prism here. And so you see all the different colors of light, different wavelengths are being spread out. And what we can do with this prism is we can mount it on a little camshaft and rotate that prism around. So by rotating that prism around, we're able to sweep the different colors of light back and forth over this exit slit so to select the particular wavelength that we want. So this part of the instrument is called the monochromator. So mono means one, chrome for color. So it's kind of a one color maker. So this lets you select the wavelength that you want. It shines that particular wavelength then through your sample, so we would have a sample holder. Some of that light gets absorbed, so I've drawn kind of a faint dotted line here of light coming out. A little bit of that light comes out and then hits our detector, which is a device that is able to convert the amount of light intensity into an electrical signal that we can then amplify and measure. So we call that our detector. So this is the schematic diagram. Let's go and actually look at how all these pieces work together in an actual instrument. So this is not a Spectronic 20 spectrophotometer, but it has all of the basic components that are built into the Spec 20. So first we're going to look at the source. So this is the source, and I've disassembled it a little bit so you can see that the source is really just a plain old light bulb. And there's a switch that you can use to turn that on and off. And boy, is that bright. Okay, so we're going to turn that off. The source would typically be mounted in a housing like this. And inside this housing, there's a little lens my finger is pointing at. And that lens will help focus the light or help all the light rays to go kind of in a straight line. So I'm going to reassemble this a little bit, and then we'll go and look at the next component. The next part of the spectrophotometer is the monochromator. So that's this box right here. I have my light source back here. You can kind of see that there's a little bit of light coming in here, and this is where the lens is. So light is coming from my source and going into the monochromator. So this monochromator has a lid that I can open so we can look in and see some of the guts. So if I look closely in this region, and let me see if I can get my laser pointer in there. So right here where my laser is pointing, kind of in that region, that's where the lens is coming out. And then this right here, you may not be able to see it very well. Let me see if I can zoom in on it a little bit. So there's a little bit of distortion here, but there's a little slit here. So we have a slit here that allows light from our source to be selected in just sort of a narrow band. And then this light shines on a little piece of glass, looks like glass, right here in this region where my laser pointer is pointing. And that's actually what's called a diffraction grating. So if you've ever looked at the back side of a DVD or a CD, I know that those are kind of not popular as much anymore, but you'll know that when you shine them at light, you can sort of see the rainbow spread out. Well, that's the same thing that's going on here. We have a reflective surface that has grooves cut in it, and through some very fascinating physics, optics, those grooves cause the light to be dispersed into the rainbow pattern that we're used to seeing. So there's a knob right here that's marked off in wavelength units, and as I turn that knob, it moves a little camshaft, which changes the angle that this piece of metal with the glass on it is hitting that slit. And then it goes through a couple of lenses, and over here there is an exit slit. So you can't quite see that exit slit in my video, but trust me that there's a little slit there. Now what you can see in this video is the little rainbow. So you see the rainbow off to the side there. It's off to one side, so it's right here is my little rainbow pattern. And as I adjust this wavelength selector, you see that I sweep the wavelength, the rainbow pattern, over the slit here, selecting a particular color that comes out. We can see that more clearly if I have something white-ish. We can see that as I tune this wavelength, sweeping it over the slit, that the color of light that comes out the other side is changing. Let's look at that over here on this piece of paper. So on this piece of paper, you can see that I have a color of light selected, and as I turn the knob, different colors of light will come out of my monochromator. So I'm selecting a particular color of light to send through the sample. So right here is my monochromator. This is where the sample cell would sit. And then on the other side of the sample cell is our detector. So our detector has a couple of knobs on it that we can use to adjust things. And then on this side of the detector, there's a little lens that will help focus the light onto the detector element. So if we open this box up a little bit, we can see that there are a lot of electronics. But the important component is this one right here, which I'm going to pull out. Maybe. So this little guy is called a phototube. And so you can't see it very well, but there's a little half-moon shaped piece of metal that's here in the back. So if I turn it this way, it's kind of coiled up like a tin can cut in half. So the surface of that metal, this inside surface right here, is coated with a chemical that we call photoemissive. So when you hit it with light of the right color, it will eject little electrons. Those electrons then are attracted to little metal rods that are here on the sides, and those electrons then generate a little electrical current, which we can then measure. So that's the guts of the detector. And so then as we measure that electrical current, it comes out onto our little meter here and will deflect this pointer depending on how much light is shining on my phototube. And so that's how a spectrophotometer, a Spec 20, sort of works, all the guts of the beast. Now let's go learn how to operate a Spec. So this is the Spectronic 20 spectrophotometer. This is an older model that has an analog meter reading on it. So if you're lucky, your instrument might even have a digital readout. Also, if you're lucky, your instrument will have a set of instructions printed next to it, somewhere mounted on it, so you can always refer to these, but I'm going to go through and talk about how to operate this instrument. So first, of course, make sure that it is plugged into the wall. Then you'll notice that there are two knobs in the front, one knob on the top, and then there's a little hatch that opens and closes on the top of the instrument. So the first thing that you want to do is the two knobs. You want to pick the one on the left, and it usually is labeled 0% T. Let me see if I can focus on that. No, I can't. 0% T. And so what you're going to do is turn that knob. You'll hear a click and some humming. That means it's on. Also, you'll notice that this needle might start to move on my meter up here. Good. So it needs to turn on and warm up for about 15 minutes for the light bulb and all the electronics to come up to temperature. Then the first thing that you're going to do is set the wavelength. So that's the knob on the top, conveniently labeled wavelength. And then you see there's a reading over here. As we turn the knob, I will select different wavelengths. So to show you that that's actually causing something to happen, I have a sample cell that has a piece of white chalk in it, cut so that we can see the color of light coming through. So if we look down in this hole, you will see that as I change, raise that up a little bit, so that as I change the wavelength selector, I'm getting different colors of light. Look at that. So there's orange, red, and I can change and get green, and then I'm moving more towards the blues. Pretty dim in the blue section, so you don't get to see as much of that light, but you get the idea. Alright, so you will want to set your wavelength. Listen to what your instructor tells you for setting that wavelength to the right value. So in some spectrophotometers, you should check, there is a filter down here on one side. So this filter says that for wavelengths in the range of 340 to 599 nanometers, there's a little lever under here that you need to flip so that it is underneath that range. If instead you're measuring something between 600 and 950, you need to flip the lever so that it is underneath that wavelength range. So make sure that that is set correctly. Set your wavelength correctly to the desired wavelength. So I'm going to set mine at 500. And then you're ready to begin setting up the instrument. So we've done the first two steps. We've turned it on. We've set the wavelength. Then we need to select the filter position. So our next step is to set the 0% T. So we're going to use this knob right down here, the one on the left. It's labeled 0% T in case you forget. Now with the hatch closed and no sample tube in the holder, what we want to do is adjust this knob so that our meter reads 0% transmittance. Without a sample, the light path is blocked in the Spectronic 20, and so we should be getting no light onto our photo tube. And so any current that we see is just background current, and we need to tell the instrument that that is true. So we're going to adjust this knob, and as you move the knob, you'll be able to see that the position of the needle moves. And you want to set it so that it is at 0% T. Notice when you look at this meter scale that the top scale is measuring % T, the bottom scale is measuring absorbance. So we want to read the % T scale, the top scale. Now when you go to set 0% T and you're reading the scale, you want to make sure that your eye is positioned directly in front of the needle so that you can read this scale with precision. This particular scale helps you to do that because behind the scale there's a little mirror that reflects the pointer. You want to hold your eye so that you cannot see the reflection of the pointer in the mirror. So move your eye back and forth so that you cannot see the reflection of the pointer in the mirror. And then adjust your knob so that it is right at 0% T. So you will need to get a sample holder, a sample cell. Sample cells in spectrophotometry are called cuvettes. Now this one looks a lot like a test tube, but it is way more expensive than a test tube. So your instructor will appreciate it if you are careful with these. So you'll notice that it has a little white line marking right here. The reason that they're so much more expensive is their internal diameters are carefully controlled to be the same length. So it makes sense that if you were to expand or contract the distance of this tube a little bit, your samples would absorb more or less light because your sample light is traveling through a longer path. So these cuvettes are expensive. They have exactly 1 centimeter of path length, and this little line here is to help you put it into the spectrophotometer in the same orientation each time. So you're going to take one of your cuvettes and you're going to fill it with what we call a blank solution. So a blank solution is technically a solution that contains everything except the molecule that is your particular absorber. So very often that will simply be distilled water. So you can fill your test tube with distilled or deionized water, and then you're going to load it into the sample cell. So I will show you how to do that. So you're going to lift the hatch. If you look really closely here, there's a little raised line right there. We always want to make sure we put the cuvette in the same way each time. So we want to line up that line with the white marking that is on our cuvette. So we're going to point that so that it is lined up with this little line right here, and we put our cuvette into the sample cell. So you'll want to find a cuvette, and you'll want to fill it at least half full with deionized water, or whatever your blank solution is. Then, because we may have been handling this test tube, we may have gotten fingerprints on it, so we'll want to wipe those off. We do that by using a chem wipe, which is a lint-free piece of tissue paper. So I will do that, take out a chem wipe, and I'm going to wipe the outside of this tube so that it is free of dust, fingerprints, and all of that sort of stuff. You will also want to make sure that there are no air bubbles inside your test tube that might scatter light. If that's the case, then a gentle thump might help to dislodge them. So make sure your tube is wiped off well. Now you're ready to insert your blank solution. So open the hatch. Make sure that you are lining up your white line with the little mark that is on the sample holder. So we're going to insert that into the instrument, and you're going to push it all the way down. Not too hard, you don't want to break the test tube. And then close the hatch. And now what we're going to do is set 100% T using this right hand knob. So the left hand knob is for 0%, and the right hand knob is marked 100% T. So that's step 6, set 100% T or 0 absorbance. So we're going to turn this knob so that the needle is sitting exactly on the opposite end of the scale, right at 100% transmittance. So right now we don't have anything in our solution that should be absorbing light at 500 nanometers wavelength. So 100% of that light should be getting through to our phototube. So we have now blanked our spectrophotometer. Now normally you won't need to recheck those, but it's not a bad idea to periodically check and make sure that if you put your blank solution back in or have no solution in, that it's reading 0 and 100% T. So over the course of a lab afternoon, those tend to drift. So now we're going to remove our sample. So to prepare your sample, you could use either a separate cuvette, but it's good practice to actually use the same cuvette to measure both your blank and your samples. Although normally people will think it's handy to have a separate tube for their blank and a tube for their sample. But best practice is to use the same test tube for all of your measurements. That way, if there's slight variations in the manufacture of the test tube, you will have everything calibrated to calibrate those out. So that's the most precise way. So I'm going to dump this deionized water into this waste beaker right here. And now I'm ready to fill it up with my sample. But you'll notice there are drips of water left on the inside of my cuvette, so I need to wash those out. So it is good practice to always rinse your cuvette 2 or 3 times with some of the sample that you will be measuring so that you do not dilute it with drops left behind from your previously contained solution. So I'm going to rinse that a couple of times with a little bit of the solution that I'm going to measure. And so I'll kind of roll that between my fingers, dump it out, and last rinse. Another good practice, and oftentimes you will do this, you will measure solutions that have known concentration. We call those standard solutions. And so when you're doing that, you want to start from the most dilute to the most concentrated. That way, if there are any drips left behind, they're not going to affect the concentration of the more concentrated sample that you dump in there appreciably. So that's also good practice. So now that I've got my sample loaded, I'm ready to put it in the spectrophotometer. So we're going to go over here to the spectrophotometer, open our hatch, make sure our white mark is lined up with the notch on the sample cell holder. Push that all the way down, close the hatch, and then we should be able to measure the absorbance of the sample. So I put a dye in here that absorbs strongly at this wavelength, and almost all of the light is being absorbed. So I recommend that you read the top scale, %T. If you look at the bottom scale, absorbance is actually probably the number that we want, but you'll see that the scale is not linear, that the lines, the markings in between the tick marks, are not equally spaced. So sometimes it can be hard to figure out exactly what your absorbance reading is. So like for me, I'm somewhere in between 1.5 and 2, but I really can't tell. But up here I can get a pretty clear idea that I'm at like 1.5% T. And so I'm going to read that number. My scale is marked off in percents, 1%. So I'm going to estimate that reading to 0.1%. So I would say this is 1.5% T. So that's a pretty strong absorbance. And then you can proceed to measure the absorbance of the rest of your samples if you have more, following a similar procedure. So you're going to dump your solution out, rinse it with the next solution to measure, load it in the spectrophotometer, close the hatch, and record the %T. You can then use that equation that I showed you to convert %T into absorbance units, which is going to be linearly related to concentration. So you'll want to find a cuvette, and you'll want to fill it at least half full with deionized water, or whatever your blank solution is. Then, because we may have been handling this test tube, we may have gotten fingerprints on it, so we'll want to wipe those off. We do that by using a Chem wipe, which is a lint-free piece of tissue paper. So I will do that, take out a Chem wipe, and I'm going to wipe the outside of this tube so that it is free of dust, fingerprints, and all that sort of stuff. You will also want to make sure that there are no air bubbles inside your test tube that might scatter light. If that's the case, then a gentle thump might help to dislodge them. So make sure your tube is wiped off well. Now you're ready to insert your blank solution. So open the hatch. Make sure that you are lining up your white line with the little mark that is on the sample holder. So we're going to insert that into the instrument, and you're going to push it all the way down. Not too hard, you don't want to break the test tube. And then close the hatch. And now what we're going to do is set 100% T using this right hand knob. So the left hand knob is for 0%, and the right hand knob is marked 100% T. So that's step 6, set 100% T or 0 absorbance. So we're going to turn this knob, and we're going to watch the needle move. And this needle is moving kind of slowly, but it's getting there. So I had my filter in the wrong place. I told you guys to check that earlier, but I forgot to do that. Alright, so we're going to adjust this so that the needle is sitting exactly on the opposite end of the scale, right at 100% transmittance. So right now we don't have anything in our solution that should be absorbing light at 500 nanometers wavelength. So 100% of that light should be getting through to our phototube. So we have now blanked our spectrophotometer. Now normally you won't need to recheck those, but it's not a bad idea to periodically check and make sure that if you put your blank solution back in or have no solution in, that it's reading 0 and 100% T. So over the course of a lab afternoon, those tend to drift. So now we're going to remove our sample. Now we're ready to measure our sample. So we're able to insert our sample. So let me talk about how to do that.